This is the first recording for Chapter 7, the Muscular System. In this chapter, we will be discussing the functions, structures, and features of skeletal muscle fibers, and discuss the neuromuscular junction at a skeletal muscle fiber. We then discuss coordinated contractions and how ATP plays a role in muscle contractions. Then we have one slide on muscle fiber type and conditioning. And then we have two slides, one for cardiac muscle and one describing smooth muscle. Last, we discuss some muscle terminology and then do a tour of the muscles of the human body, starting with the axial skeletal muscles and the appendicular muscles. The 5 skeletal muscle functions are listed here on this slide. So let's talk about each one. Skeletal muscles produce movement of the skeleton by pulling on tendons that then move bones. These muscles are responsible for maintaining posture and body position. Skeletal muscles also support soft tissues with the muscles of the abdominal wall and the muscles of the pelvic floor. Skeletal muscles guard entrances and exits in the form of sphincters, and skeletal muscles maintain body temperature. When contractions occur, energy is used, and that energy, some of it, is dissipated as heat. And that's how we can maintain our constant body temperature. Skeletal muscles are organs that contain 3 layers of connective tissue. The epimysium covers the entire muscle. The perimysium divides the muscles into bundles called fascicles, and blood vessels and nerves are contained in the perimysium. The endomysium covers each muscle fiber and ties fibers together. It contains capillaries and nerve tissue. Where the ends of all 3 layers of connective tissue come together and attach to a bone, that forms a tendon. Similar to tendons, aponeuroses are a broad sheet of collagen fibers that connect muscle to each other, but do not necessarily connect bone to muscle. Blood vessels form an extensive network in skeletal muscle, and these are going to provide a high amount of nutrients and oxygen to skeletal muscles, which have very high metabolic needs. Nerves are mostly under involuntary control, so they are stimulated by the central nervous system via axons, which push through the epimysium, branch through the perimysium, and enter the endomysium. In skeletal muscle tissue, single skeletal muscle cells are also called skeletal muscle fibers. So those terms can be used interchangeably. This figure shows the organization of those structures that I just mentioned. Notice if you take an entire muscle organ here from the forearm and enlarge it, you can see the tendon, which is that collection of the epimysium, perimysium, and endomysium. Looking a little bit closer at that organ, at a cross section of that muscle organ, you can then see the endomysium as this yellowy color along the outside. The perimysium is this grayish color, and then the endomysium is on the inside of one of these muscle fibers. If we were to take one of these muscle fascicles and enlarge it, that's what we would have right here. So this is a bundle of muscle fibers. So the perimysium here is bundling those muscle fibers together. And you can see the endomysium is highlighted here as the pink layer between each one of these muscle fibers. So if we were to take a closer look at this muscle fiber, that's what we would have down here. Remember, the term muscle fiber is interchangeable with a skeletal muscle cell. Those two terms are the same. Okay. And you can see myofibrils as these red dots here. Again, this is a cross section. Notice the mitochondrion and the nucleus. Here is an axon of a neuron, and we have a capillary here as well. Here, the only connective tissue that is visible at this magnification is the endomysium. Now, something I want to point out to you is that the cytoplasm of a muscle fiber, of a muscle cell, that cytoplasm has a special name, and that's called the sarcoplasm, pictured here as kind of a light pink. Also, The muscle fiber, or a skeletal muscle cell, has a special cell membrane, which then gets a special name, the sarcolemma. And those are denoted here on this slide. So you're probably noticing then that these muscle fibers are quite different from a typical cell. They're specifically organized to produce contraction, and they have specific names for general cell structures. These skeletal muscle fibers can be very long and are typically multinucleated. If we bounce back to that previous picture, looking at the muscle fiber, here's a nucleus and here's a nucleus. So they're multinucleated. And they are composed of highly organized structures, giving them a striped or a striated appearance. And we're going to talk about that in a moment. The special name of muscle fiber plasma membrane is the sarcolemma, which has openings across the surface that lead to a network of transverse tubules, or T-tubules. The T-tubules allow for electrical stimuli to reach deeper into each fiber. The sarcoplasm is a specific name for the muscle fiber cytoplasm. And inside each muscle fiber, branches of T-tubules are going to encircle cylindrical structures called myofibrils. Myofibrils. So myofibrils are bundles of thick and thin myofilaments consisting mainly of actin, which are molecules found in thin filaments, and myosin, molecules that are found in thick filaments. So these are the contractile proteins that are shortened and are responsible for contraction. The sarcoplasmic reticulum is a specialized smooth endoplasmic reticulum that forms a tubular network around each myofibril. It contains high concentrations of calcium ions, which are released during a contraction. Also, a triad is a combination of two terminal cisternae and one T-tubule. So that sarcoplasmic reticulum is expanded at the end, called a terminal cisternae. And that's easier to see on this picture here. Again, I want to point out the name of the special cell membrane around this skeletal muscle cell has its own name, the sarcolemma. The sarcoplasmic reticulum is that special endoplasmic reticulum around a myofibril. Notice how it has this enlargement at the end. This is the terminal cisterna. And T-tubules, which are parts or channels of the sarcolemma, are going to lead in between and throughout and kind of interculate between these myofibrils. So taking a look at a myofibril is made up of myofilaments. You have the thin filament and the thick filament. The thin filaments are pictured here in pink or darker red, and this thick filament is pictured in purple. Notice the mitochondria, which is embedded, multiple mitochondria embedded in that skeletal muscle fiber. This is a nice figure showing the thin myofilaments made of actin. So actin consists of a thin twisted protein with specific active sites for myosin to bind to. At rest, these active sites are covered by strands of tropomyosin, and they're held in place by troponin. This is an illustration of one of the thick myofilaments, which are made of myosin. A thick filament, myosin, has a tail and a globular head. And this globular head is where the myosin can attach to active sites on actin during a contraction. The smallest functional unit of a skeletal muscle fiber is called a sarcomere. And this is formed by repeating myofilaments arrangements, and each myofibril has about 10,000 sarcomeres, so very, very small, packed into these myofilaments and myofibrils. The thick and thin filaments are arranged and they produce a striated appearance of the fiber. Overlapping filaments are going to define lines and bands. We have four important lines and bands: the Z line, M line, A band, and I band. And let me tell you about those while looking at this picture. The Z line is shown here in pink, and it does zigzag back and forth to make what looks like the letter Z. So this Z line has the thin actin filaments, and it forms the end of the sarcomere. So from one Z line to the next Z line is an entire sarcomere. Another protein is going to connect the Z lines, that's this little green one right here, to the thick filaments to maintain an alignment of all of those myofilaments. The M line is this nice straight line right here, colored in purple for you to see. The M line is made of connections between the thick myosin filaments. The A band, pictured here as this region and pulled out, separated from the rest of the sarcomere. The A band contains all of the thick filaments, and the I band, from here to here, pulled out for you here, stretched out. The I band contains the thin filaments and includes the Z line. So briefly, all these structures come together in a muscle contraction, and as you can see, calcium really is the key to this contraction. So first you have calcium released from the sarcoplasmic reticulum, and that calcium will bind to the troponin on the actin. This will cause the troponin to change shape, and the tropomyosin, which is connected to the troponin, will move away from the active sites. It exposes the active sites of the actin. Once those active sites of the actin are exposed, then myosin heads are going to bind to those active sites and create cross bridges. And this will cause a rotation and cause the actin to slide over the myosin. So when a sarcomere contracts, the I bands get smaller, Z lines move closer together, and the H bands decrease. The A band doesn't change, which indicates that the thin filaments are sliding towards the center. So this is the sliding filament theory, and I have a nice video to illustrate this contraction at the sarcomere level. I'm just going to zoom in a little bit here.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.
So you have access to this video to watch as many times as you'd like. All you have to do is go into Blackboard and download the PowerPoint and click on that hyperlink, and it will take you to this site where you can play this video as many times as you please. And so that brings us to the first checkpoint of this chapter. And I ask you to describe the basic structure of a sarcomere, and that's a very ambiguous type of a request, but your answer should include the A band, the M line, the Z line, etc., etc. So not just composed of thick and thin filaments. We're going to need a little bit more of a precise answer to that question. And then you can start the next recording for this chapter after you answer that checkpoint.